My grandmother, God rest her soul, she used to have this saying she'd drop on me whenever I was panicking about a deadline or, I don't know, lost set of keys.
Ooh, yeah.
Yeah, she'd always say, honey, don't sweat the small stuff.
It's a classic. I mean, it is great advice for keeping your blood pressure down.
Oh, absolutely. Terrible advice for biology, though.
The absolute worst.
Right.
Yeah. In our line of work, if you stop sweating the small stuff, you basically miss the entire point of existence.
Exactly. And today, for this deep dive, we are taking Grandma's advice and just throwing it right out the window.
Good call.
Because we are sweating the absolute smallest stuff imaginable. We're talking about the bustling, just chaotic rush hour traffic that is happening inside your cells right now as we speak.
It is surprisingly violent in there, isn't it?
It really is.
We tend to have this mental image of a human cell as this peaceful, sort of watery balloon floating in space.
Yeah, like a spa day for mitochondria.
Right. But if you actually zoom in, it's like Times Square on New Year's Eve.
Or the Tokyo subway during the morning rush. It is just packed.
You've got proteins bumping into organelles, motors hauling cargo, chemical signals flying everywhere. It's a literal mosh pit.
And imagine trying to find one specific person in that mosh pit.
In the dark.
In the dark. And not just find them, but watch exactly what they're doing.
Like, is he tying his shoe? Is he texting?
Right, and doing it without bumping into him or changing his behavior at all.
Yeah. And usually, to see anything in the dark, you need a flashlight. But in biology, your flashlight keeps running out of batteries every 3 seconds.
Or, to make the light last, you have to strap a generator the size of a refrigerator to that person's back.
Just to see them.
Which naturally makes it really hard for them to dance in the mosh pit.
Exactly. That is the fundamental catch-22 of bioimaging that scientists have been just banging their heads against for decades.
Well, welcome to the Deep Dive Lab, everyone. Because today we are looking at a breakthrough that claims to have actually solved this impossible trade-off.
It's really exciting stuff.
We're talking about a paper published in Nature Photonics in October 2025 that might just change how we watch the movies of life.
Yeah, this is some really cutting-edge work coming out of the Chinese Academy of Sciences and their associated institutes.
Yes, specifically from lead authors Hongwei Yang and Zikuan Yan.
Right.
They worked with a larger team including Yefei Jiang, Qingrui Fan, Jianjun Wang, and Xiaohong Fang. And the title of this paper is Brace Yourself. Single-chain ultrasmall fluorescent polymer dots enable nanometer-resolution, cellular imaging, and single protein tracking.
It's quite a mouthful.
It is, but they developed something with a very catchy name.
SUPdots.
SUPdots. It sounds like a candy or, I don't know, a new app.
Technically, it stands for single-chain ultra-small fluorescent polymer dots.
Which is why we're definitely sticking with SUPdots. The mission for this deep dive is to understand how these tiny little specks of light are basically upgrading our view of life.
Right, from a grainy 1980 security camera to full 4K resolution.
And doing it without requiring $1 million budget, which is a key point we really need to get to later.
Absolutely. That's the best part.
Okay, so let's unpack this. We need to understand the old world before we appreciate the new world.
Right.
Why is it so hard to watch a protein? I mean, we've had microscopes since the 1600s. Why can't I just zoom in and watch a motor protein take a walk?
Well, it's not just about magnification, it's about contrast. Proteins don't naturally glow.
Right.
If you look at a living cell under a light microscope, it's mostly just transparent slush. To see a specific protein, say a kinesin motor, walking along a track, you have to tag it.
Attach something to it that emits light.
Exactly.
Okay. And we had fluorescent tags for a long time. I remember learning about GFP in high school biology.
Green fluorescent protein.
Yeah, the glowing jellyfish stuff.
Right. And fluorescent proteins like GFP are great because they are small. They're usually roughly the size of the protein you're tagging, so about 3 to 5 nanometers.
So it's like a biological lantern.
It is. But it has 2 huge problems. 1, it's not that bright, and 2, it bleaches.
It bleaches, like leaving a t-shirt out in the sun.
In a photochemical sense, yeah. When you hit these organic molecules with the intense laser of a microscope to make them glow, they break down.
Oh, I see.
They fade out very quickly. You might get a few seconds of video, and then poof, the light goes out.
So you completely lose your subject in the crowd, just as they're doing something interesting.
Exactly. So to fix that, material scientists invented synthetic nanoparticles, things like quantum dots.
Okay.
These are amazing in terms of light. They're super bright, and they almost never fade. You can blast them with lasers all day.
So problem solved, right? Just use quantum dots.
If only. The problem is the size.
Huh.
To make quantum dots stable in a biological environment, they end up being relatively large.
How large?
Often 15, 20, sometimes up to 30 nanometers wide.
Now, I have to play devil's advocate here for a second. To me, and probably to you listening, 20 nanometers sounds incredibly small. A human hair is, what, 100,000 nanometers wide?
Yeah, in our everyday world, 20 nanometers is totally invisible. But in the protein world, 20 nanometers is an absolute monster.
Right.
If your target protein is only 5 nanometers wide and you attach a 20 nanometer tag to it.
Oh, that's the refrigerator analogy.
It is. It's exactly like strapping a refrigerator to a marathon runner. In chemistry, it's called spheric hindrance.
Spheric hindrance.
That's right. The tag physically gets in the way. It bumps into other structures. It creates drag. So if you use a quantum dot, are you actually watching the natural movement of the protein?
No, you're watching a protein struggling to drag a heavy backpack.
Precisely. You're watching a strained system.
So we've basically been stuck between a rock and a hard place. Either we use a small tag that goes dark too fast or a bright tag that's just too big and clumsy.
Yes. And that is exactly where the team from the Chinese Academy of Sciences enters the chat. They asked, can we make something that is as small as a protein under 5 nanometers, but as bright and tough as a quantum dot?
Enter the subdot.
Enter the subdot.
So chemically speaking, what is this thing? Is it glass? Is it metal?
It's a polymer, essentially a conjugated plastic. Now, usually when you make polymer dots, you run into a huge manufacturing headache.
Which is?
Okay, imagine spaghetti.
I'm always ready to imagine spaghetti.
Picture a pot of boiling spaghetti. The noodles are the long polymer chains. They're floating around in the water, which is our solvent. Now, if you want to make a particle out of that, you usually evaporate the solvent. You drain the water.
Okay.
What happens to the hot spaghetti noodles as the water vanishes?
They clump. They just stick together into a giant starchy ball.
Exactly. In chemistry, we call that multi-chain entanglement. As the solvent leaves, the chains grab onto each other. You don't get a single noodle, you get a meatball.
And that meatball is a large nanoparticle. Too big for what we want.
Right. So you end up with the bulky backpack again.
So the researchers needed a way to freeze the spaghetti noodles individually, mid-air, before they could touch each other and tangle up.
Yes. And this brings us to my absolute favorite part of the paper. The technique they used is called splat-cooling.
Splat-cooling.
Yeah.
It is a fantastic name. Very descriptive.
It really is.
It sounds like a move from a slapstick cartoon, but the physics are actually really sophisticated.
Yeah.
Walk us through the splat.
So they take a very dilute solution where the spaghetti noodles are floating really far apart from each other. Then they drop tiny droplets of this solution onto a surface that is frozen at minus 150 degrees Celsius.
Minus 150. That's colder than the surface of Mars at night.
It's extreme. When the droplet hits that surface splat, it undergoes something called vitrification. It freezes instantly.
Instantly.
It happens so fast that the polymer chains literally do not have time to find each other. They are frozen in place as individual chains.
You know, it reminds me of that scene in X-Men where Quicksilver runs around and everything else is just frozen in time. The water droplets are just hanging there in the air. They just pause the chaos.
That is a perfect visual. They freeze the chaos of the solution. Then they use a process called freeze drying or lyophilizing to sublime the ice away.
Which means turning the ice directly into gas.
Right. And what's left behind are these tiny, individual, untangled polymer coils.
Single-chain, ultra-small fluorescent polymer dots.
SUPdots. Exactly. Because they aren't tangled with other chains, they remain tiny, under 5 nanometers.
Okay, so they've made the tiny tag. They've solved the size issue. But does it work? Is it actually better than the refrigerator backpack?
The data is honestly staggering. They tested a few different types of polymers, some glow blue, some green, some red.
And they gave them catchy acronyms like PFO and CNPV. But let's stick to the performance. How bright are we talking?
They measured the photon count, literally counting the particles of light coming off this thing. The CNPPV subdots were tracking roughly 1.4 times 10 to the power of 4 photons per second.
Which means?
In plain English, they are about 15 times brighter than standard quantum dots or dyes of similar colors.
15 times brighter. That's not a marginal gain. That is the difference between a birthday candle and a spotlight.
And remember, they're doing this while being a fraction of the size. That's the magic of the splat. By keeping the chain isolated, it prevents the polymers from quenching or dampening each other's light.
Wow. Now here's where it gets really interesting for me. The paper mentioned that these dots have, like, personalities.
They do. They found that different polymers behave differently. The PFO and PFBT types had what we call blinking behavior. They turn on and off.
Like a digital signal, 1, 0, on, off.
Exactly. But the other types, CNPPV and PDFDP, they glowed continuously. They call that analog behavior.
Why does that matter, though? Why would a scientist want a blinking light? Wouldn't that be super annoying?
It depends entirely on what you're trying to measure. If you're trying to count exactly how many proteins are in a cluster, blinking is actually really helpful. You can just count the blinks.
Oh, I see.
But if you're trying to track movement, you want a continuous light so you don't lose the track. The fact that suPdots offer both options is a huge advantage.
So you can basically pick the right light bulb for the job.
Exactly.
Okay, let's talk about what they actually saw with these things. Because they didn't just make the dots in a vacuum, they took them for a test drive inside actual cells.
They did. They started with something called clathrin-coated pits.
Clathrin-coated pits. CCPs, these are like the cargo bays of the cell, right?
Right. They're these little structures on the cell membrane that help bring nutrients into the cell. Now, under a normal confocal microscope, a CCP just looks like a fuzzy blob. It's too small to see the details.
It's like looking at a blurry photo of a tire. You see a black circle, but you don't see the treads or the hubcap.
Good comparison. The researchers used suPdots with a technique called STED microscopy. STED is a type of super-resolution imaging. It breaks the diffraction limit of light.
And when they turned on the STED with the suPdots, what happened to the blob?
The blob resolved into a donut. A continuous hollow ring structure. They could clearly see the empty space in the middle.
Wow.
And they measured the diameter of these rings at about 178 nanometers.
And that number is important.
It's crucial because that matches the data we have from electron microscopy.
Okay.
Electron microscopes are great for detail, but you can't use them on living things because the sample has to be dead and frozen.
Right.
The fact that the suPdots matched the electron microscope measurements proves that the suPdots weren't distorting the structure. They were small enough to coat the ring perfectly without making it look puffy or fake.
So they saw the donuts.
Mm-hmm.
But the real showstopper, the climax of this paper, in my opinion, was the walking protein.
Ah, yes, kinesin 1.
This is my absolute favorite protein. If you've ever seen those animations online of a little stick figure dragging a giant bubble along a tightrope that's kinesin, it's the delivery truck of the cell.
It is remarkably human-like in how it moves. It literally takes steps along microtubules. Left foot, right foot.
But these steps are tiny.
They are incredibly tiny. And tracking those steps inside a living cell is sort of the holy grail of microscopy. Usually, to see steps that small, you need a machine called MMFLUX.
Which I assume is not something you just pick up at the local hardware store.
Definitely not. It's a $1 million instrument. Very complex, very rare. Most labs do not have one.
So the challenge here was, can we see kinesin walking in a living cell using standard equipment just by upgrading the tag to a suPdot?
That was the bet. They expressed kinesin 1 in HeLa cells, which is a standard line of research cells, and tagged them with suPdots. Then they used a spinning disk microscope.
Okay.
That's a high-end machine, but it's standard. It's the workhorse found in many university basements.
Yes.
And they saw it.
They saw the footsteps.
They captured the kinase moving at 50 frames per second, or 50 hertz, and when they analyzed the data, they could see distinct steps.
How big was the step?
They measured it at exactly 15.7 nanometers, roughly 16 nanometers.
And that's accurate.
It's spot on. That is exactly the step size of kinesin 1 known from structural biology. But previously, seeing that required slowing the protein down or using that $1 million MFLUX machine. They did it at full speed, in a living cell, with a standard scope.
That is just mind-blowing. It's like filming a flea hopping on a dog from a mile away and being able to measure the exact length of the hop.
And doing it with a home camcorder instead of the Hubble telescope.
Right, that's the democratization part we talked about.
Exactly. It means you don't need the most expensive machine in the world to do cutting-edge physics. You just need better shoes on the runner. In this case, suPdots.
And because the suPdots are so stable, they could watch it for a while.
Yes. Normal dyes would have bleached out in seconds. The suPdots kept glowing, allowing them to track the movement over time. This is critical because biological processes aren't snapshots, they are movies.
Right, you need to see the whole sequence to understand what's happening.
It really changes the game from taking a picture to watching a documentary.
That's a great way to put it. So let's bring it all together. What does this all mean for us? We've got these tiny bright dots.
To summarize, the team created suPdots fluorescent probes under 5 nanometers. They completely solved the bulky backpack problem.
And they did it with that cool splat-cooling method, freezing the spaghetti before it tangles.
Which is a massive manufacturing breakthrough in itself. And the result is a probe that is bright enough and small enough to track the tiniest machinery of life, like motor proteins, in real time, in living cells, without needing exotic equipment.
It really feels like we're finally upgrading our security cameras.
We are. For decades, we've been looking at cellular biology through a blurry black and white security feed. SuPdots are like upgrading to 4K color video.
And when you can see better, you understand better.
Think about diseases. Alzheimer's involves proteins clumping together. Cancer involves signals going haywire and cells moving where they shouldn't. Viruses hijack these motor proteins to travel to the nucleus.
Oh, wow. I hadn't even thought about that application.
Yeah, if we can watch these processes step by step, literally nanometer by nanometer, we can understand exactly where things go wrong. It takes the guesswork out of the mechanism.
It turns biology from a what science into a how science?
Exactly. And SuPdots are a tool that makes that accessible to so many more researchers. It's not just for the elite, well-funded labs anymore.
That is a fantastic takeaway. Well, I want to thank everyone for stopping by the Deep Dive Lab today. We've gone from rush hour in Tokyo to freezing time in X-Men, all to watch a little protein take a 16-nanometer step.
It's been quite a journey.
If you enjoyed this deep dive into the nano world, make sure you subscribe to the channel. We love decoding these breakthroughs for you.
Always a pleasure to explore the cutting edge.
Before we go, I want to leave you with a thought. We talked about watching kinesin walk today, but SuPdots could theoretically be used to tag almost anything. So here is my question for you listening. If you could use these SuPdots to zoom in and watch any process inside your own body happening live, like your neurons firing a specific memory, or your immune system fighting off a cold virus in real time, what would you choose to watch?
That is a really fascinating question. Seeing a memory form, that would be something else.
Right. Think about it and let us know. Until next time, keep sweating the small stuff.
Because that's where the magic happens.
See you next time.